My name's Paul Howes. I'm the manager for the Centre for Sustainable Aquatic Research. I manage the facilities. We have around 15 different recirculating aquaculture systems and other aquatic research laboratories. I thought it would be a great opportunity today for you to come and see each of the different rooms that we have here. We have recirculating systems that have a range from being very small practical systems that we can use on a day-to-day basis for smaller, shorter projects, all the way up to industry scale aquatic recirculating systems that are up to around 60 cubic meters in size. Welcome to the first room in Caesar. This is the largest room that we have within the facilities. So this is the room RAS B, so it's a recirculating aquaculture system B. This is where we do most of our commercial work in terms of large numbers of fish, large numbers of production, high densities of tanks. This room has 12 1,500 litre tanks and 9 3 cubic metre tanks. This allows us to have many fish being able to be grown within these systems, which allows us to compare any of the results or the experiments that we're doing with real commercial levels and numbers of fish. So as you can see within these systems currently, the tanks are in the stage of just being cleaned, ready for the new system. And now we have different batches of lumpfish eggs incubating in these incubators. Within this particular system, over the last 4 or 5 years, we've produced close to 3 million lumpfish within these facilities, within this one particular system. Many of them very small, but some of them larger deployment size fish which get sent up to the salmon cages in Scotland. This room is one of our new rooms that have been funded through the Smart Aqua project. This particular room is a twin system recirculating aquaculture system. As you can see, there's 12 tanks on either system. This allows us to modify or change either the salinity, the pH, or the turbidity, or the flow rates within the systems, so we can fine-tune how the fish respond to different parameters that they may be exposed to, and how that would therefore affect things like feed conversion rates and the growth of the fish as well. This is our ornamentals lab. The aim is to have a range of different marine ornamentals within these systems, where we're going to be looking at different feeding types, different feed trials, and how we can boost and benefit the welfare and the quality of the fish that are being raised within the ornamentals industry in the UK. We'll be looking at how different parameters can affect the colour and the quality of the fish that are being produced within the marine ornamental industry. Now we're entering the CRISPR laboratory. This is a laboratory where we focus on gene editing. We have a range of different species that we work with here. All of them are animal models, such as the zebrafish. We also have killifish, which are able to clone themselves. And we also are about to acquire some turquoise killifish as well. We look at a range of different studies here, so studies to do with longevity and aging, studies to do with obesity, studies to do with diet requirements, and these species can act as a model for similar mechanisms that would happen within humans. Okay, now we're moving to some of the medium-sized systems that we have within Caesar. Within this laboratory, we look at how different trophic levels interact with each other, and the main aim of working with the aquaponics area here is that we have a range of species of fish we can hold in tanks or filter feeders, and we can also look at how the waste from these particular species can grow different plants. Okay, so we're now entering C2. This is a room that we use for innovation, so it's our innovations lab. We work with a range of different industry partners within this room, as well as a number of different students who do short trials in order to answer questions for industry. You can see that we have a range of different tanks. They're all in multiple numbers, so lots of duplicates and replications for getting decent quality statistical data out the other end of the trial. Okay, so now we're entering a lab that looks quite different to some of the other labs we've been in previously. These particular laboratories are designed specifically for model organisms again, so zebrafish this time. As you can see, they're in a rack formation with multiple small tanks. This again allows for a lot of replication. Within this room, we're concentrating on improving and standardizing the welfare of these particular species that are now one of the most commonly used organisms within animal research for model organisms for humans. This particular room is C4. This is the microalgae lab. Within this laboratory, we hold our main master cultures. So we have 25 plus different master cultures, some of which we've had for 30, 40, 50 years. Each week they need to be re-inoculated, and from these master cultures we can increase the size quite rapidly through to small beakers, then to carboids. From carboids they can go into these bags, and from the bags we can move them out to the biofences that you'll see later on within the facilities. So we're now entering tropical lab. As you can see, the temperatures are really quite high. It's kept at around 28 degrees water, and the air temperature is around 25 degrees as well. So it's a pleasant place to work during wintertime especially. Within the tropical lab, the system is set up to have the full life cycle of tropical species. So we have broodstock tanks here, we have the larval tanks, we have the fry tanks, and it's all controlled again by a main recirculating aquaculture system. So this particular building here is a building that's being created specifically to look at the different trophic levels within aquaculture and how they interact with each other. The unique and novel part about this building is that we have a glass house built onto the end of these two main recirculating aquaculture systems, and within the glass house we have a PBR which allows us to grow microalgae based on the nutrients that are produced from the waste from the fish. The main aim of this is to try and look at ways to capture opportunities within the circular economy, that is, by which we can have waste streams and make the waste streams themselves a way to generate new income or new products within the circular economy.